DNA structure and replication. What is DNA? You might wonder. DNA stands for deoxyribonucleic acid, carries genetic information that determines traits, found in the nucleus of eukaryotic cells, discovered by Watson and Crick, 1953, based on Rosalind Franklin's X-ray data. Components of DNA. DNA is made of nucleotides, each with 1. Deoxyribose sugar, 2. Phosphate group, 3. Nitrogenous base, 4 bases, adenine A, thymine T, cytosine C, guanine G. The A base pairing rules. 2 strands twisted into a double helix. C pairs with G, 3 hydrogen bonds. Backbone, sugar and phosphate. Rungs, base pairs. A pairs with T, 2 hydrogen bonds. DNA organization in cells. DNA coils around histone proteins, forming nucleosomes. Nucleosomes coil into chromatin fibers. Chromatin condenses into chromosomes during cell division. Process of copying DNA before cell division occurs during S phase of cell cycle. Ensures each new cell gets an identical copy. Steps in DNA replication. 1. Initiation. Helicase unwinds DNA. 2. Elongation. DNA polymerase adds nucleotides, leading and lagging strands. 3. Termination. Ligase joins Okazaki fragments, forming 2 identical DNA molecules. Enzymes involved. A helicase unwinds DNA then. Primase synthesizes RNA primer then. DNA polymerase adds nucleotides then. Ligase joins fragments then. Topoisomerase relieves tension ahead of fork. Semi-conservative replication. Each new DNA molecule has 1 old and 1 new strand, and confirmed by Messelson and Stahl, 1958. Importance of DNA replication. Essential for growth, repair, and reproduction. Errors can cause mutations leading to disorders or evolution. And the summary. References. Watson and Crick, 1953, Nature. Messelson and Stahl, 1958, PNAS. Campbell Biology, 12th edition.